Hello and welcome to this week's featured video. Today we will explore group comparison of endpoints in Simulx, which is an important tool in clinical trial simulations. Every decision made during this process can significantly influence the outcomes. So it is important to base these decisions not only on graphs and charts, but also on information that we can really trust. Imagine we are designing a clinical trial to compare a new drug with a current standard of care. Treatment efficacy is evaluated by measuring changes from the baseline of a specific PD variable. Here we have output distribution for both treatments, with the reference group on the left and the test group on the right. We see differences between them, but they do not automatically guarantee the success of the clinical trial. We have to prove that the effect of the new drug is statistically superior to that of the reference treatment. And this is where Simulx group comparison feature comes in. It really helps to see if differences that we observe are meaningful or not. The group comparison is located in the Outcomes and Endpoints section. You can activate it using the group comparison toggle, and you can choose between direct comparison or a statistical test. If there are multiple endpoints, each one can be customized separately. In this video, we will explore the methodologies behind this feature and show how it works. This video is a continuation of the series of videos dedicated to outcomes and endpoints. So if you're new to these concepts, I recommend watching the previous videos first. Group comparison offers many benefits. For example, it enables us to identify if a simulated treatment has a statistically significant impact on the endpoint. We can quantify the magnitude of differences between endpoint values, which is a critical step to determine the clinical significance of the results. Furthermore, we can analyze variability in treatment effects among groups and verify if it can be used by a broader population. We can also quantify how changes in various parameters impact the treatment effect. It gives a better understanding of what can happen in different scenarios. Group comparison helps also to find optimal factors such as sample size, patient selection criteria, and which endpoints to focus on. We will see an example of this application in the end of this video. Now, when we better understand the importance of group comparison, let's jump into details of how it works. When you activate the group comparison toggle, one of the simulation groups defined in the simulation block must be set as the reference group. Other groups are then compared to it. For each endpoint, you can choose between a direct comparison of endpoint values or a statistical test. Let's start with a direct comparison. It uses mathematical operators to compare endpoint values directly against a user-defined value. In this example, my endpoint is an arithmetic mean of a change from the baseline of a PD variable. By default, the comparison checks if the means in the reference group and test groups are only different. However, this can be changed to a statement that the mean in the test group is greater than the mean in the reference group, and adding a threshold value to the difference, such as a half a unit, allows us to check if the endpoints differ by at least this value. When this logical comparison is true, the trial simulation is considered successful. It is important to note that the direct comparison that we describe here does not take into account the number of individuals in each group. It only compares endpoint values. However, in most cases, we not only want to check if there is a difference between the endpoints, but also if this difference is statistically significant. To do this, we need to consider both the magnitude of the difference and the number of individuals over which the endpoints are calculated. And this can be done with statistical tests. Statistical tests begin with formulation of relevant hypotheses for the chosen endpoint. In the example from the introduction, our goal is to demonstrate that the effect of the test drug is significantly greater than the effect of the reference treatment. To do a statistical test, we first state the null hypothesis, which represents the opposite of what we want to prove. So, the null hypothesis is that the reference treatment is better than the test treatment. The statistical test will verify if this null hypothesis is likely or not. If it is sufficiently unlikely, for example, if its probability of being true is below 5%, then the null hypothesis can be rejected in favor of the alternative hypothesis, suggesting that the test drug is indeed superior and consequently the difference in means is greater than 0. This is what we want to prove in the statistical test in Simulx. Similar to direct comparison, statistical tests also use different mathematical operators. The non-equal sign corresponds to a two-sided test. For instance, we will use it if we wish to show that the mean in the test group only differs from the mean in the reference group, while operators larger and smaller give single-sided tests. For instance, to check that the mean in the test group is significantly greater than the mean in the reference group. The statistical test results in a p-value, which is compared to a user-defined threshold. By default, this threshold is set to 0.05. If the calculated p-value falls below the threshold, then the probability of the null hypothesis is below 5%. In this case, we can reject this hypothesis in favor of the alternative hypothesis. In this example, we would conclude that the mean in the test group is significantly greater than the mean in the reference group. Group comparison is run together with outcomes and endpoints, and the results are automatically displayed in the Results tab in the Group Comparison section. The table contains the difference between the reference and test group, p-values in case of statistical tests, and information if the trial is successful or not. In the case of replicates, each replicate is represented on a separate line. The Summary tab provides the percentage of successful trials over replicates. It is the power of a trial. In the presented example, there is a 76% probability of demonstrating that the mean in the test group is greater than the mean in the reference group. Difference of means is one of the possibilities in group comparison. It depends on the type of the endpoint. You can find a complete guide in our online documentation on the Simulx webpage. For continuous outcomes, statistical tests use the difference of medians, arithmetic means, or the ratio of means if the endpoint is a geometric mean. In case of ratios, the null hypothesis states that the ratio is 1, contrary to the difference where it was set to 0. For time to event outcomes, statistical tests use the difference of median survival between test and reference group. And for binary outcomes, where endpoints describe the percentage of true responses, statistical tests use an odds ratio between the test group and the reference group. The odds ratio definition is different if you use paired samples, which is in the case when groups have the same individuals, or unpaired samples. But you can find all the formulas in the documentation. Type of statistical test also depends on the type of an endpoint and the option same individuals among groups. As promised, let's go back to our example where we aim to determine if the mean for the test drug is significantly greater than the mean of the reference drug. The percentage of successful trials is 76%. It is obtained with a sample size of 40 individuals per group. Increasing it to 100 improves the trial's power, which now achieves a success rate of 92%. A common question is to find an optimal sample size to have at least, for example, 85% chance of success. We can automate this search using lixoftConnectors. because the 2023 version of Simulx has new functions. We can define outcomes and endpoints, get their values, and run group comparison. In the online documentation, in the section with examples on how to use these functions, you will find a sample code to run a simulation with different sample size and plot the power of the study based on an endpoint. In our example, the group size of 80 individuals per group is sufficient to achieve 85% probability of a trial success. Thank you for your attention and have a good week.